So today we're going to be talking about, or continue talking about eukaryotic gene regulation, and we'll start with talking about at the mRNA level. So previously we had talked about regulation of the initiation of transcription, but what about after transcription? What about after the RNA is already made? Well, you can remove RNAs from the cell through the use of something that we call RNAi. Now, RNAi has different types of RNAs associated with it. MicroRNAs, which are abbreviated as MI for microRNAs, and small interfering RNAs, or siRNAs. These are single-stranded RNAs that can bind to the mRNA that they are complementary to, so you can target a specific mRNA that you want to knock down. And proteins, additional proteins are going to come in and degrade that mRNA. So if that mRNA is not around, it can't be translated into a protein. So let's look at this a little bit more closely. So the primary RNA transcript can actually have a whole bunch of these hairpin structures in it. That's due to, this is what we call secondary structure, when a single-stranded RNA can take on shape, because the regions in the middle here are complementary to the regions across from them. And so it can form these structures. Now, this is the microRNA that we're going to take a closer look at over here. So initially, this would be cleaved off of this precursor, this primary microRNA transcript, and now we have this shorter hairpin. This enzyme here, called dicer, is going to cleave the ends off of the hairpin, so now we just have a double-stranded RNA. So it's not DNA, this is all RNA. Now, the proteins that are going to form the RISC complex, that stands for RNA-induced silencing complex. So there's a whole bunch of proteins here that come together with one strand from this double-stranded RNA. So this is our microRNA. When it comes together with that single-stranded RNA, we form the RISC complex. Now, the RISC complex uses that microRNA. Okay, so the microRNA is part of the RISC complex, but it uses it to find mRNAs that have the complementary sequence. And when it finds them and binds to them, the RISC complex is going to degrade those mRNAs so they can no longer be used for translation. So here's our little ribosome guy down here. So if there's no mRNA for that gene, we're not going to get any protein. Now, microRNAs that we just looked at in the previous slide are naturally occurring. They're found inside of cells, but we can take advantage of this natural process and apply it to scientific experiments and potentially therapy. So what you can do is you can genetically engineer a plasmid that contains a sequence to create a short hairpin RNA. So they call it shRNA, short hairpin RNA, when you introduce it artificially through a plasmid. So that's SH right here is for short hairpin. It's going to be transcribed. Once you force it inside cells, that plasmid is going to get transcribed. And then because it forms this hairpin here, it'll be recognized by dicer. It gets cleaved, turned into an siRNA that can then bind to the proteins to form the RISC complex. The active RISC complex is once it has the single-stranded RNA that was encoded initially in this plasmid here. And it can seek out and target the mRNAs that you want to knock down. A knockdown is when you target the RNAs and knock them down so they can't be expressed. A knockout is when you remove the gene entirely from a cell, but that's a different topic. So if you want to know what a gene does, you prevent it from functioning. So this is one potential way that you can do that. You can knock down the mRNAs and see what are the effects on the cell when you remove the ability to make that protein. So we've talked about the ability to remove mRNAs so that the protein's not made in the first place, but what about if the protein is already made? Do we still have any control? And the answer is yes. There are still several levels of control here. Now, one thing you might want to do, let's say you already made a bunch of this protein, but now you don't need it anymore, so you want to get rid of it. There are little proteins called ubiquitin. They're these little green guys here. Ubiquitous means kind of like found everywhere. And when they discovered the protein, they didn't know what it did, but it was found everywhere, so they named it ubiquitin. Turns out that if you tag a protein, the protein here, the one we're looking at is the purple one. Ooh, look at these alpha helices. Secondary protein structure. But if you tag a ubiquitin molecule on there, it actually takes a couple, getting them on there. It targets this protein to the proteasome. The proteasome is itself a protein complex. It's a barrel shape that murders proteins. So it's going to break our little purple protein down. It's going to break those peptide bonds, and they're going to be released as amino acids that can be used to build a different protein. Or it could be broken down to short peptides that can then get recycled to amino acids. So that's one way to control expression of a gene, like at the protein, or control how much protein is present. That's not the only way to control proteins. Some proteins require cleavage. That means that they have a certain section of the protein that makes them inactive. And if you want to activate them, you need a protease. A protease is an enzyme that can cleave part of a protein, so that's how you would activate those proteins. The protease cleaves off the little part of it that prevents it from working. Now you have an active protein. We've talked before, remember we talked about kinases can add phosphate groups to proteins. Phosphatases can remove phosphate groups or dephosphorylate them. Usually if you are adding a phosphate group to a protein, you're activating it. If you're removing that phosphate or dephosphorylating, then you're usually inactivating it. Now, if you remember, the prefix glyco means sugar. Like when we talked about glycolysis means breaking sugar. If you are glycosylating something, you are adding a sugar. Some proteins need sugars to be added to them in order for them to function. That would happen in the Golgi. Remember we talked about the Golgi apparatus is where packaging happens. Packaging is like, you know, if you're giving a Christmas present to somebody, you're going to package it, you're going to wrap it, make it look real pretty, maybe slap a bow on top. That's kind of like glycosylation. You slap a sugar on top of that protein and it's going to function. So let's take another quick look here at how we can control gene expression or regulate gene expression in eukaryotic cells. Remember, we have chromatin modification. Methylation closes up the DNA and makes it into heterochromatin. Acetylation allows those nucleosomes to move apart and allow for transcription to occur. Transcription, remember which activator proteins are present to bind the enhancer elements, determines which genes get turned on in different cells. Remember that it folds over and creates this hairpin shape here. Okay, and so different cell types would have different activator proteins present that would activate like different enhancers and turn on different genes because each gene has different enhancers associated with it. RNA processing, we have alternative splicing. We also have RNAi. Now, RNAi can actually be involved in chromatin modification as well. So those small non-coding RNAs, sometimes you'll see that abbreviated as ncRNA. There's actually tons of different RNAi that we don't even understand yet going on, but those can actually influence chromatin modification as well. Translation, RNAi can prevent translation, right? Don't forget that you also have all those ways of controlling the function of the protein itself. So how active it is. Does it need a sugar group? Does it need to be cleaved by a protease? Does it require a phosphate group? Can you target it for degradation by adding some ubiquitin proteins onto it? Yep, this stuff here is all that I just said. There's also proteins that regulate the activity of the ribosome and the access of the ribosome to the mRNA. So that's another opportunity that you have to like slow down or speed up translation. So the actual making of the protein. Also remember that even without RNAi, mRNAs are unstable molecules. Remember that they have that extra oxygen in their sugar in ribose. It's not found in deoxyribose, the sugar of DNA. That makes it unstable and it's liable to fall apart. So having different length 3' Poly A tails can make them last longer or shorter, so it will eventually degrade on its own. That's all I have to say about that. Make sure you take good notes on this and that you can identify different ways of controlling how much protein is present and how active it is.